Synaptic transmission is the process by which one neuron communicates with another neuron or a target cell, such as a muscle or gland cell. It's a complex process involving several key steps. 1. Neurotransmitter synthesis and storage. Neurotransmitters, the chemical messengers of the nervous system, are synthesized within the presynaptic neuron. These are then packaged into small vesicles, which are stored near the presynaptic membrane. 2. Action potential arrival and depolarization. An action potential, an electrical signal, travels down the axon of the presynaptic neuron and reaches the axon terminal. This depolarizes the axon terminal membrane. 3. Voltage-gated calcium channels open. The depolarization opens voltage-gated calcium channels in the presynaptic membrane. Calcium ions Ca² rush into the axon terminal, triggering the release of neurotransmitters. 4. Exocytosis of neurotransmitters. The influx of Ca² causes the vesicles containing neurotransmitters to fuse with the presynaptic membrane. This process, called exocytosis, releases the neurotransmitters into the synaptic cleft, the small gap between the presynaptic and postsynaptic neurons. 5. Neurotransmitter diffusion and binding. Neurotransmitters diffuse across the synaptic cleft and bind to specific receptor proteins on the postsynaptic membrane. These receptors are often ligand-gated ion channels. 6. Postsynaptic potential. The binding of neurotransmitters opens or closes ion channels in the postsynaptic membrane, altering its permeability to ions like sodium, not potassium K, or chloride Cl. This creates a postsynaptic potential (PSP), an excitatory postsynaptic potential (EPSP) if the membrane depolarizes, or an inhibitory postsynaptic potential (IPSP) if it hyperpolarizes. 7. Signal integration and termination. Multiple PSPs are integrated at the postsynaptic neuron. If the overall depolarization reaches the threshold potential, it triggers an action potential in the postsynaptic neuron. Neurotransmitter action is terminated through reuptake by the presynaptic neuron, enzymatic degradation in the synaptic cleft, or diffusion away from the synapse. In summary, synaptic transmission is a precisely orchestrated process involving the synthesis, storage, release, binding, and removal of neurotransmitters. This process is crucial for communication between neurons and the proper functioning of the nervous system. The details can vary depending on the specific type of synapse and neurotransmitters involved. Furthermore, research continues to uncover the intricacies of this fundamental biological process.